Okay. So you can go directly in the PubMed, you can find out the articles, you can read about it, what people are doing, because the designing of the experiment in animal cell culture is much more sophisticated and delicate than the plant tissue culture. Okay. And the benefit here that you have everything declared and shared with you, right? So you don't struggle with anything else. Okay. So everything basically is here. You buy it and start doing the day you receive. Okay, so that is fantastic. If just going to here, if you want to find out the bacteria, for example, application, cell model, collection, herd, animal cell, microbial. No. It must be E. coli. Okay, let me find out. Okay, they have the whole collection of the microbes. I know. I think they have separated the bank or don't know what is happening. Microbe products. You see here, Amelia? Microbe?
Yes.
Yes, you click on it and you have all the bacteria, the fungi and different kinds of viruses, even they sell you viruses and different kind of protistos. What is this? Protistology. Should be the parasites, something like that. Okay, we can go here and see. They have anaerobics, bacteriophage, drug-resistant bacteria, extromophils, and media reagent related with them, nucleic acid, polysaccharide toxin, perceptible culture. Okay, so we can go here, anaerobes, right? And you will have here the thousands of bacteria which are authentic and you can buy in culture with whole process and protocol. Okay, they will give you everything. Okay, so that is why this place, this website is fantastic. Okay, do you see here? You have all kind of bacteria, anaerobics. If you want to put E. coli, for example, E. coli, you will find hundreds of different the strains and all that. Okay, even genetically modified, even altered, genetically modified, which are producing certain kinds of enzymes. Everything you will find. Okay. Okay, I think I'm not searching it correctly, but you know how to search, right? Here you can go and explore it, and you can go with the anaerobes and you will have it. Okay, because it is E. coli is not anaerobe, that's why it's not coming, right? But it is aerobic, no? Okay,
maybe so it could be in the bacterial resistant
sorry,
I don't know. Maybe it could be in the drug resistant bacteria.
Oh, it could be in everything, right? It is going to be everywhere. You just put here in the microbial product like this, and directly you can find out here any bacteria. Okay, it should be searching here. This is the bacteria.
Oh, okay.
19,000 bacteria. Just imagine 19,605 different kinds of bichitos, right? Everything here. And here you will find even the what kind of susceptibility, what kind of resistance, what kind of antibiotic, which country, location, extromophilus, type or strain. Here E. coli, 998 E. coli available. Do you see this? Okay. Are we okay?
I have a question.
Okay. Yes, go ahead.
I have two questions actually. The first one is, if you are working with virus, well, if you want to order a virus, do you have like to have a specific permit or something so that it can send that to you?
Yes, of course. Until unless it is not going to be, until unless it is BSL2 lab, right? If it is BSL1, BSL1 is the simplest one. So you can take it, you can bring it. But BSL2 is for many viruses, right? So whenever it is BSL2, I will not take it. I will send it to some of my collaborator labs. There we will work on it, right?
But I mean, how do they know if you are capacitado to work with that?
No, they don't know. They don't care. That is up to you. You are the one who is responsible for this.
Okay.
Okay. They cannot ask anyone or everyone to show your lab and capacity. They will just, if it is because they don't sell very deadly viruses. They don't sell it, right? They don't sell anthrax. They don't sell which are very rare or viruses used for the BSL4. Okay, they don't sell it. That is prohibited. They will sell you BSL-1 mostly, BSL-2 a lot, and BSL-3 very, very limited. Okay. So obviously they can ask you once you say if they know, because BSL-3 labs are very limited, right? So they have whole data that how many BSL-3 labs are in Mexico. So maybe they can inquire you, but they are not always looking after, you know, every buyer. Okay, but definitely they will poke you. Okay, BSL-3, BSL-3, BSL-3. Okay, dangerous, something like that. So it is depending on the buyer.
Okay, thank you. And the other question is, how much do they have to send about the quantity of cells they send you? Is there like
oh, in every protocol on the site, in the product information, they mentioned. Okay, you can go. I showed that they gave you the aliquot quantity. They told you you have like 10 to the power fourth cells per milliliter. Okay, so by that you can calculate how many virus you are going to have, how many is supposed to survive the transportation. So they gave you the enough amount, and they will tell you how many milliliter you have to take it out to put and create your own culture. Okay.
Oh, yes, thank you. Thank you.
And they have a very nice call center service. If you have any problem, you can call them. The technical expert committee, they can guide you for many purposes.
Thank you, teacher.
You're welcome. Yes, Carla.
But like, for example, in the overall picture, like they are not expensive, right? I know the prices are like really high, like if you wanted to casually buy one cell line, but like the prices are nice. Oh, okay.
Yes, obviously they are very, very affordable. Okay, they don't do much business about it, right? This is, they promote our research and they self-finance themselves. This is just for maintenance, sustainable project. But they are very affordable. A bacteria E. coli you can buy in 5,000 pesos. Okay, a good cell line. I'm going to order 4 cell lines now, and the cost is like 40 to 50,000 pesos. Okay, depending on the cell line, depending on the specificity. Okay, one cell line may cost you sometimes 50,000 pesos also, or another cell line may cost you only $200. Okay, so depending on the availability, depending on the precision and specificity of each cell line. All products are different, but they are not in $1 million. Okay, they are not very, very costly. They are the normal cost for research. Okay, cool. So now I'm moving for the another topic. Hope this ATCC thing is clear, because okay, let's do one thing. How would I do this? Okay, I need that every one of you, not everyone, every team of you enter in the ATCC, or without entering ATCC. I need you to, but basically the protocol and everything has to be helped by ATCC. I need every team of this group, all groups, right? All 5 groups, please define a cell line, okay? A cell line, and define the protocol for this, okay? Very similar way you did for the plant tissue culture, but this is going to be very, very different, right? So here in one page, okay, in one page, or maybe 2, because maybe you are going to directly copy paste from the ATC data and all that. So, okay, so maybe 2 pages fine, but please try to find out the cell line of your choice, and let me know today itself by remind, okay, that this is your chosen cell line. And for the Monday or Tuesday, I need you to submit the protocol of the establishment of the cell line of your choice. Okay, how you are going to establish the cell line condition for your cell line of choice. Okay, so that is your task. So you need to decide your cell line. I had my proposal, but I think it would be better that you choose your cell lines of your choice. Okay, so find out anything, COVID-19, virus, HIV, whatever, NTREG, whatever you can think you want to study, you want to, and the objective, that is very important. Selecting a cell line for what you are going to do. Okay, toxicity testing, expression of proteins, production of monoclonal antibody, whatever it is, whatever you want to do, okay? Biotherapeutic production, whatever it is, or just establishment of the cell line to study medicinal plant, whatever it is, just define the objective and define the cell line, only one per team, okay? And then you are going to create the protocol, whole protocol very, very systematically. What you will do, I will go to the ATCC and will buy this is the catalog number, this is the instruction, these are the protocol to establish, and after the establishment, what are the things that I'm supposed to do, okay? So everything the before and everything after, okay? So at least I can understand now that now you know how to establish the whole process of animal cell culture by selecting the media, by selecting the conditions, protocol, parameters, and objective. Okay? Yes, Emilia.
Teacher, this is in groups, right? In our groups.
Sorry, it is in group. It is group, not individual. It is in group.
Okay, thank you.
Okay, okay, cool. Okay, so I need to move forward. Okay, coming to the next point. Okay, coming to next point. I would put a video directly. Okay, let's watch a small video. I am not sure if you have watched it because I have uploaded a lot of material for the hemocytometer and for the for other topics, previous topics, but I'm not sure if you are watching this. Okay, okay, let me check if it's the right one. No, this is not. Okay. Hemocytometer. This is the second video. Okay, let's do it. Let me share the sound. Or here I go. Just start with this video. Are you watching this video?
Yes.
Yes.
The hemocytometer. It's used for counting cells and was originally used for counting blood cells. Hemocytometer. Blood cell measurer. Counter. The hemocytometer is a thick microscope slide with 2 counting chambers on reflective sections. Each chamber is divided into 9 main squares with a series of etched lines. These squares are 1 millimeter by 1 millimeter or 1 millimeter squared. When a glass cover slip is placed on top of this chamber, each of these 9 squares contains 100 nanoliters of liquid. To start, you add 10 microliters of a well-mixed cell suspension to the edge of the cover slip, where capillary action sucks it up under the cover slip. I'm using a mixture of small beads to demonstrate. You then look at the grid under the microscope and count the number of cells in a square. How do you count a cell if it lies right on the edge of the square? Well, you decide before you count on 2 sides of the square that will be considered in and 2 that will be considered out, so that you won't double count an edge cell. You do this for at least 4 squares and then average your counts together. Since you know that each of these squares is 100 nanoliters, you now have your cells per 100 nanoliter density. You can then multiply this by 10,000 to get cells per milliliter. If the mixture is very dense, you can use some of the smaller subdivided squares to help count and keep track of your cells. A useful tool for counting cells. Click here to subscribe for more. What is this thing?
Okay. And one more. Okay. Very small, but very, very.
Teacher, later can you send the link to the girls' channel?
It is only in the module 11, dear Carla.
Okay, thank you, teacher.
It is already there since 2 weeks, and I'm sure nobody me hizo caso. Okay, anyways, coming to this one, okay? This is very, very important. Please pay attention because this is the calculation, okay? This is very important, and we will do right away after this video.
Simplified microscope slide containing 2 identical wells or chambers into which a small volume of cell suspension is pipetted. We have already removed 100 microliters of our cell suspension and placed it in a microcentrifuge tube. Dilute the suspension by adding 100 microliters of trypan blue. Trypan blue is a dye that helps us distinguish between living and dead cells. The dye passes through the membranes of dead cells, so they will appear blue under a microscope. Living cells exclude and will appear mostly clear. Load both chambers by pipetting the suspension under the cover slip. Now place the hemocytometer under the microscope. Each chamber is divided into a grid pattern consisting of 9 large squares. Each square has the same dimensions and contains 10 to the -4th power milliliters of suspension. The rules for counting cells sometimes differ from lab to lab. In our lab, we count cells in the 4 large corner squares and the center square. Let's count the cells in the first square. 1, 2, 3, 4, 5, 6, 7, 8. So what about the cells that are touching the outside boundaries of the square? In our lab, we count the cells that touch the top and left boundaries, and we ignore the cells that touch the right and bottom boundaries. 9, 10. We need to count the number of both living and dead cells. Remember, the dead cells will appear blue. Occasionally, you will see artifacts, objects or debris that appear blurry and don't have a well-defined shape. This is an example of an artifact. We won't include it in our count. Proper storage, cleaning, and handling of the hemocytometer will minimize the number of artifacts. There are 10 viable cells and 1 non-viable cell in the first square. Now, the top right square. There are 9 viable cells and no non-viable cells. Next, let's count the bottom right square. There are 11 viable cells and no non-viable cells. And now the bottom left square. There are 10 viable cells and 2 non-viable cells. And finally, let's count the cells in the center square. Sometimes cells will appear as clumps or small groups. It may be difficult to determine exactly how many cells are in a group. The method of counting clumps of cells differs from lab to lab, so be sure to follow the procedure in your lab. We will count this clump as 2 cells. There are 14 viable cells and no non-viable cells in the center square. The total number of viable or living cells from all 5 squares is 54. The total number of non-viable cells is 3. Now that we have counted our cells, there are several calculations we need to perform. First, let's calculate the percentage of viable cells. Here's the formula. 54 viable cells divided by 57, the total number of cells, gives us 0.947. Multiply by 100 and the percentage of viable cells is 94.7%. Next, let's determine the average number of cells per square. We counted 54 viable cells. We divide 54 by 5 because we counted in 5 squares. The average number of cells per square is 10.8 cells. Now let's calculate the dilution factor. The dilution equals the final volume divided by the volume of cells. Our final volume is 200 microliters because we started with 100 microliters of cells and added another 100 microliters of trypan blue. 200 divided by 100 is 2. Therefore, the dilution factor is 2. Next, we need to calculate the concentration of viable cells, the number of living cells per milliliter. Our average count per square is 10.8. The dilution factor is 2. 10.8 times 2 times 10 to the fourth power equals 216,000 cells per milliliter. We can write the concentration using scientific notation as 2.16 times 10 to the fifth power cells per milliliter. From our calculations, we now know the concentration of cells in our culture is 216,000 cells per milliliter, and approximately 94.7% are viable living cells.
How's that? Clear? Okay.
I like that video.
Yes, it's very nice. Okay, so let's do a small exercise. You all can open your app that you downloaded. Remember that the name? Hemocytometer, Sidekick. You have like two ways, okay? Let's do one thing. We can do it manually, like the calculation we have just seen, okay? Or you can do it by this application that I told you to download. You go to the home, and below the home you have an icon that is called Hemocytometer. Okay, click on it, and you will find basically the. Let me remove my background. Okay, please open your app. I hope everybody has downloaded, right? How to remove my background? Okay. Okay. So you have now, you're seeing my screen. You have basically the Hemocytometer copy on your screen, right? So this application is going to help you that it is on your table when you are observing your hemocytometer by the microscope. Okay, so let's do one thing. This is going to be the second part, and start from the first one. As I mentioned that we have two ways to do this, manually or automatically, right? So in this case, the most important thing that you have already know that the structure, the 1 millimeter square grid structure, 9 big squares, and within the 9 you have 16 small squares like that. Okay, so first, so what we can do is you also know the rule that you avoid counting the cells which are on the right and the bottom line, right? And you also know that the dilutions, okay, because most of the time, or sometimes, okay, at least most of the time you use the 100 microliter of the Trifon blue, that is a dye, right? Trifon blue, and many people use it. Many people use it because they want to check it out, okay? Or sometimes you don't use it at all. So that your dilution factor is going to be only 100, okay? 100 microliters. So basically, your dilution factor here in this case is going to be 1, and in this case it's going to be 2, okay? Suppose if we are going to count it, so this is the protocol, okay? I will, this is already I uploaded. So first of all, you need to harvest the cell line, right? You put the 100 microliter of the cells to the hemocytometer, okay? And depending, in this case, if I'm using only this one, my dilution factor is only 1. If I'm going to add additional 100 microliter of Trifon blue, then it is going to be 200 microliter of the solution, right? So my dilution factor is going to be 2, okay? So one very important thing, you can use even only one square if you are in a hurry and if you have a very long experiment, because it will give you at least some approximate number of your cell line. But when you want to have the more precise number of the cell lines, then you have to go at least with 4 circles. So when you go with the 4 circle, just add one more. So the 5 circle strategy is the best one and the most solid information you will get. Okay, so suppose we have these this is square, right? Square number 1, square number 2, square number 3, and square number 4, and this is the final one. Okay, so you can count cell here, you can count cell here, you can count cell here, you can count cell here. Let's put one example. You have like 10 cell here, okay? You have another 10 cell here, you have another 10 cell here, another 10 cell here, okay? Out of the 10 cell, how many dead and live cells? Suppose we have 10 cell live cells and 2 non-viable cells. Here in 10 cells, you have 3 non-viable cells. Here you have like 10 viable cells and 0 non-viable cell. Here you have 10 viable cell, like 2 non-viable cell. Okay, so you have to count the total number of viable cells, okay, and total number of non-viable cells. Okay, that you can count manually, but the app can help you because you can just click on the block and put 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, and this is how you can just register your cell line. Oh, sorry, cell number on this. After when you have the basic information about viable and non-viable cells, you can go with this calculation. Let's do it our own. For example, let's open the calculator without touching the application. Okay, right now, let's do it. For example, the first circle, okay, we need total viable cells, total number of viable cells, total number of non-viable cells. Okay, so let's take out our own number. What are the total number cells? The total number of viable cells, 10 plus 10 plus 10 plus 10 plus 40, right? So let's put 40. We have 40 viable cells, okay? Now, how many non-viable cells? We have 2 plus 3 plus 2 plus 0, okay? Oh, sorry. So we have 3 to 5 to 7 non-viable cells, okay? So what is the total number of your cells? Is 47. Are we okay here? Are you following me? Yes?
Yes, teacher.
Yes, teacher.
Yeah.
Okay, so the number one formula, first formula is number of viable cells divided by the total number of cells. So let's divide. We have number of viable cells 40, and this will be divided by the total number of cells, that is 47. Okay, you will calculate and divide by 100. Okay, let's divide it. 40 divided by 47 equal 0.85 multiplied by 100. Okay, so we have 85%, 85.10% of viable cells. Are we okay here? This is our percentage. Okay, then take out the second calculation, that is average number of cell per square. So this is the viable cell number is 40, and the squares, how many square did we use, guys?
4.
4. Good. Okay, so you divide it, and very simple, you have 10. Okay, average number of cells, which is 10 per square. Okay, now go with the calculation number 3, which is calculating the dilution factor. So what was the final volume, guys, in our solution? What would we do? How could you identify the viable and non-viable cell? By adding trifon blue, right? So what would be our dilution final volume? 200 or 100?
200, right?
200, yes. So this is the 200. Our total volume was 200 microliter, and that has to be divided by the volume of cells, that is 100 microliter, which is you taken out from your chambers or your flask. Okay, so divided by 200 divided by 100, and you have your dilution factor, which is 2. Last calculation, the concentration of viable cells, that has to be estimated by multiplying average number of cell per square plus dilution factors divided by 10 to the power 4. Okay, this is the dilution, that is the dilution volume that we have mentioned there. Okay, so now, for example, here, what is your percentage? The average number of cell per square. So here we will put 10, right? Your dilution factor 2, okay, divided by 10, 4. Okay, what is the calculation? Tell me.
200,000.
Yes. Yeah. Are we okay? 2,000, 200,000 is okay?
Yes.
Yes? Good. So now you can put it like 2 multiplied by 10 to the power 5. Okay. So you can put it like scientifically 2.0 multiply by 10 to the power 5 cells per milliliter. Okay. Isn't it simple? It looks a little complicated, but it's very simple. Just you need the first number of your cells. That's it. After that, the calculation can be done very, very easily. Are we okay till here? Hello, hello.
Yes.
Yes.
Yes, okay.
Okay. Now come to the app. Okay. For example, I was doing the exercise here, but I did it here. So basically the same. Okay. So you can do it on the presentation. Okay. You can do it here. I just prepared it that way. Okay. So this is like 40 by 47. Number of cells is like 10. Dilution factor is 2, right? 10. And what was the number I forget here? The 47, right? So you have 47 here, and that's it. You can divide, and finally you can divide it. You have the 2.0. Okay, multiply by 10 to the power 5 of cells. Okay, cells per milliliter. Okay, that is why you are putting this thing to convert it into the milliliter. Okay. Now let's do it by the applications. Are you on the app? Are you on the app? Okay. So just imagine that you are with your microscope. Okay. So you have like this option. You have basically your hemocytometer here. Many people do so many repetitions to make a very precise number, but we will go with only one. Okay. So for example, just click on minus. There is an option here. Okay. This is the minus number. You see it here, blue. Click on it. So you will have only one grade. Okay. One square. Click on it, please. Okay. I will click. Look how it would convert. 1, 2, 3. Okay. So now you have the whole hemocytometer and your blocks. Then now just you have to count your cells with this finger. Okay. Just with this finger, the red one. Okay. Suppose in this well, number 1, how many cells you see? I see 7 cells. 1, 2, 3, 4, 5, 6, 7. Okay. It will even spread like this. Okay. It will spread like this. Now coming to this one. How many cells? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay. Coming on this, I have only 1, 2, 3, 4, 5, 7. Okay. Coming to this 1, 2, 3. Just one difference in this app, you are not going to calculate only viable cells. Okay. Not dead cells. Okay. That is the point. So here 5, 4. Okay. Now you have a one button that is called calculate here at the bottom. Okay. Put on the calculate, and here you got your number of the cell.
If the last square, how much cells do you put, teacher?
You can put the, for example, it asks you the dilution factor. So if you have added the tryptophan blue, you can put 1. Sorry, you can put 2. Okay, and the chamber depth is obviously the same. Okay, 100 micrometer. So it will give you the right away the number of cell per milliliter, which is 8.25. In my case, 10 to the power 4 cells per milliliter. Okay. Can you see this? At least something you see on my screen?
No, teacher, we can see.
Now?
It's too little, but it's clear how it works.
Yes, but you have in your hand so you can test, right?
Yes.
Yes, teacher.
Yes.
Okay, if I want to add one more block, I just need to add it. I want to make the 5th square. 1, 2, 3, 4, 5, 6, 7 cells. I find out, calculate again, and it will give you the next number. Okay, so you have both options. I just want you to teach the manual one, which is very important also. Okay, and this one. This is very practical. Okay, so you have both options. You can quickly count your cells. Are we okay? And you can put the sample name, you can save it. And if you have so many petri dish, you just calculate 10, 12, 15, 18 petri dish, put your name, and it will give you the Excel chart. Okay, so you can download the Excel chart directly to your email and can add into your diary, okay, experimental diary, or in your research data. Okay, so for example, I can put anything. For example, the Vero one. Okay, I will put Vero one, return and save. Okay, so you can even add it. Okay, and you can directly email. You can directly email this information to your inbox. Okay, I can put email and it will send you the data directly. Okay, I have another app. If you want, I can tell you, but so many app will also create some problem. So this one is very, very nice, very, very practical. Here you have one more option to find out the blood differential. You see here, it has another application to find out the blood cells. Okay, how you can count, how you can differentiate them. It's very useful. Okay, this is one option. Blood differential, you have your monocyte, how they looks like and all that. So you have a very good information. You see it. So this I like it personally, and you can see on the results. This is you calculated, you already have it. Okay, all the results are stored. Okay. So hemocytometer clear. Okay, cool. So now coming to the third point, and that is hybridoma. Now, first in your seed belt. Okay, because this is a little bit more information. Okay, this is the hybridoma. Okay. Okay, are you with me?
Yes.
Yeah. Okay, so hybridoma technology. Hybridoma technology, basically the main objective is to producing monoclonal antibodies. Monoclonal antibodies, they are in the body of animals and humans all the time. Okay, and they are the response against any kind of antigen. Okay, the basic structure of monoclonal antibody is like this. It's a biostructure. It's a biostructure like this. Okay, and it has the heavy chain, okay, which is called this one. Okay, and it has a light chain. Light chains means only 2 block. Okay, and the heavy chain. So it's a biostructure, and it is the something that attach on the antigen. Okay, and these are basically produced in our body by lymphocyte naturally. Okay, they are always being produced for any kind of bacteria, virus, or pathogen, or any foreign particle enters in the body. Our cells, blood cell, produce the antibodies in response. So they can be very, very specific for one particular type of antigen. Okay, so this is, for example, and there are a variety of antibodies available naturally. Okay, and there are basically 5: IgG, IgE, IgD, immunoglobulin. Okay, when I say Ig means immunoglobulin or antibody, is the same thing, but immunoglobulin is more immunological term. Okay, so we have IgM and IgA. So they all 5 have their own source of production and source of action. Okay, IgG, for example, okay, here you can see where they are produced and what is their functions. So IgG, basically it is behind our neck. IgA again, it is in our stomach. IgA again, it is our intestine. So you will see that basically in our whole stomach, okay, the IgA antibody is the prominent one. Okay, basically anything which is related with the mucosal tissue, mucus related tissue, they are there. Okay, and other antibodies, they are in the blood. Okay, they are in circulation. They are distributed throughout the whole body. IgG, for example, it has a simple structure. IgM, it is the most complex structure. Okay, it's like a flower structure. IgA could be diverse. It could be like mixing 2 IgG together or 3 IgG together, or sometimes they will be very similar to IgA. Okay, IgD and IgE are also very similar to IgG, but still we do not know the function of IgD. Okay, IgE, we know that they have important function in the LIG reactions. IgA is basically anywhere which you have the mucosal tissue, and IgM is produced in the antigen invasion. So you remember the COVID-19, everybody is talking about IgM, IgM, IgM. You remember that? You know that? So that is the reason, because IgM is related with the foreign antigens. Many times it is a virus. Okay. IgG is the highest opsonization and neutralization activities. Okay. Opsonization and neutralization. Opsonization is attacking and neutralization is finishing. Okay. They are classified into 4 other subclasses: IgG1, G2, G3, G4. Okay, so they are more or less, this is how they are studied. Now, the monoclonal antibody, since we know that they have therapeutical natural function in our body, these 3 scientists, George Kohler, Cecil Meinstein, and Niels Jerne, they have developed a technology in 1975 for the production of hybridomas. Okay, hybridomas are the hybrid cell, very similar like a hybridoma of a protoplast, which has the capacity of producing antibody, okay, from the lymphocyte cell that naturally produce B cells in our body, okay, sorry, monoclonal antibodies in our body, and with the characteristic of the myeloma cell, which is a cancer cell which never dies, it's continuous growing, it's an immortal cell line. So it has a single cell that produces the antibodies and it will never die. So that is the hybridoma. And these 3 scientists, for this discovery, they received the Nobel Prize in Physiology and Medicine in 1984. Okay, so this was a very revolutionary breakthrough in human history, because for now antibodies, they were used in the many therapeutic applications for cancer, for sclerosis, for Alzheimer, Parkinson's, so many diseases, and even just for production of the antibodies, and just for the using for the finding out the blood relations, right? The blood group, just checking out the blood group is also based on the antibody procedures, right? So just let's little deeper. The antibody producing lymphocyte B cells are the very important cells, that are the cells which are going to produce your monoclonal antibody. Okay, so basically they are isolated from the spleen, el bazo. Okay, in Spanish I think spleen called bazo, from a mouse that has been immunized with certain kinds of antigen. Okay, so when you put any particular antigen, the spleen produce antibodies, okay, corresponding antibodies. And then we have a single myeloma cell that is from bone marrow tumor cells and capable of dividing indefinitely. It's a cancer tumor cell. Okay, so when you make a hybrid between these 2 cells, this is the product, and that is called the hybridomas. Okay, and it inherited the capacity of antibody production, and it also inherited the capacity of immortal malignant cancer cell. Okay, so that's the whole basic fundamental behind it. Now, how you do this? It has the whole procedure. Number 1, start from the immunization of mouse. This is our ratoncillo, and here we are going to inject any particular antigen. It could be even a bacteria, it could be in a virus, it could be any protein, it could be any enzyme, it could be nanoparticle, whatever it is. So for that, they will produce the antibody in their blood. Okay, after that, obviously you are going to isolate, okay, isolate these B cell from the spleen. So you have to kill the mouse, obviously, and take out this spleen out. Okay, and then this spleen will be full of the desired antibody. Okay, now on the same side, you are cultivating the myeloma cells, okay, which is the cancer cell line, and now you are going to fuse with beta cell. Okay, beta cell or B cell, not beta cell, sorry, it's B cell, B lymphocytes. Okay, and you are making a fuse. very similar thing will happen, which will be fused, which will be unfused, or badly fused or rightly fused, like very similar thing that happened in protoplast, or very similar thing that happened in the competitive cells that you make for genetic engineering lab. Okay, so finally, when you have the right cell, okay, which has the capability of both cells, then you can easily produce and harvest your product, your desired antibodies. Okay, it could be in vitro, it could be in vivo. Okay, that does not matter, but what you need a particular cell line basically. Okay, now it looks very simple, but now it has complicated things, some little complicated things. Okay, and that is about the selection procedure. For example, mixing is fine, finding a cell line is fine, finding a B cell fine, finding a myeloma cell fine, is everything fine. But how you are going to select the right fused hybridoma cells, okay? And that is the whole strategy for which they got Nobel Prize, okay? For example, and please pay attention because I think I won't have time to put the video, so that is you are supposed to watch, okay? It's a very simple video and where you can understand the whole process of selection, because it is quite confusing. Okay, first of all, myeloma cell, okay, cancer cells cannot synthesize antibodies, right, as they lack HGPRT gene. Okay, so myeloma cells does not have HGPRT gene, okay, which is required for the synthesis, for the synthesize the enzyme hypoxanthine guanine phosphoribosyl transferase. Okay, and this is the full name of this particular thing. So this myeloma cell does not have gene, and if it does not have gene, it cannot synthesize this HGPRT, okay, which is an enzyme. Okay, second thing, the process of the fusion can be done by PEG. You remember the very famous PEG or everywhere. This is the most important thing for fusion. Okay, so once you are mixing them, okay, this is the spleen cell, this is the B lymphocyte cell, this is HGPRT plus. It has the gene which is capable of producing the enzyme, and myeloma cells does not have HGPRT. Okay, now without knowing more details, you are quite confused. And for the selection of this process is depending on the hard media. It's a solid media basically, and it has a very important compound that is called hypoxanthine aminopterin thymidine, hard, okay, hard media. So this is the media, okay, it has hard, okay, and you have the both of the cells, which is HGD plus, means they produce the HGPRT enzymes, and these are the cells they do not produce it. Hope you are fine until here. Okay, now hard medium, this is why this is called hard medium, contains a drug that is called aminopterin. This drug blocks one of the two pathways that helps our cells in human and animals to produce our nucleotides, ATCG, to synthesize the DNA. We have two pathways, the cell-based pathway and de novo pathways. Okay, we have normally both of them. Okay, if one doesn't work, the second will help, de novo and salvage. Okay, these are the both pathways we have naturally in our cells to produce DNA, to synthesize DNA. So this aminopterin is a blocker that block one of the pathways, okay, for nucleotide synthesis, making the cells dependent on the another pathway that need HGPRT enzyme, okay, which is absent in myeloma cells. Okay, so myeloma cells basically are not going to survive anyhow in this media if they are not fused. That is the logic. Independently, they don't have any capacity because they don't have HGPRT gene that will not allow them to grow, because one pathway is blocked by HGPRT itself. And the second pathway will be blocked because the heart media has aminopterin. So basically, myeloma cell has no way to survive, okay, until unless they are fused with the HGPRT plus. So at least they have one pathway that can help them to survive, because another pathway is blocked by heart, okay. And finally, both of the cells, whether this one, which is HGRT plus or myeloma plus, both of them are going to die within few hours, because both of them cannot use one or another or both of the pathways of nucleotide synthesis. So forcefully, intentionally, they have to fuse to be able to survive by one of the pathways, because the another pathway is still blocked because there is a blocker that is called aminopterin. Okay, are you getting me? Are you following me? Let me see your
Yes.
face. Okay,
That's true.
you understand this?
Yes, Ashu.
Okay, fantastic. Well, just to watch it or learn it visually, I have uploaded a couple of videos. Okay, they are open. You can watch it and you can just clarify if you have any doubt. But anyways, so if you have a cell which is not going to be fused, it's going to die. That is its destiny. Unfused cell, fused spleen cell with fused, they will die. Fused myeloma cell with myeloma cell, unfused myeloma cell, they are dying. But you have a hybridoma cell, okay, that has HGPRT plus from the spleen cell, right? So they will survive because of one pathway, and another pathway will be blocked because it has aminopterin, right? So this is the simple logic, basically. Okay, so once you have selected them, basically, finally, this particular and the cell, you make a culture. Okay, now your work is create a culture. Now you can make a monolayer culture, or you can create a suspension culture depending on the particular cell lines. You remember that on ATCC you can buy directly these hybridoma cells, right? So you can buy it, they're a little expensive because they produce it. Okay, so this is the whole soul story. Aminopterin is going to block the de novo pathway. Okay, that is why they will not survive. And aminopterins inhibit the de novo pathways. Okay, so that is why the salvage pathway is the only way for the fused cells that can be survive. Okay, this is the little bit biochemical pathway here. Okay, and you can see it in details, but basically you will understand quickly, I guess. This is the de novo synthesis pathway, right? This is the de novo. Okay, and this is the salvage pathway. Okay, these are the both pathways, and our main product is production of nucleic acids. Okay, these are the nucleic acids, so they need this particular enzyme. If it is blocked, okay, they will not produce it. Okay, so this is, in this case, this is the HGPRT, right? This is very, very important. If there is no HGPRT, they will not produce a nucleic acid, right? And if they don't have, if they have aminopterin here, like the blocker, so this will block also this pathway. So basically, there is no way to create the nucleotides. Okay, and here this pathway will also be blocked. Okay, de novo pathways. So anyways, finally, this is the product. Okay, these are the cells, and this is how you are going to fuse with polyethylene glycol. And finally, you are going to have your cells, and this is how you can produce. So the application of things I will discuss in the next class. I think time is over.